Hello, welcome to another video. In this video I'm going to be explaining the lac operon and its function. So firstly we'll just talk about what the lac operon is. So it's a section of about 6,000 base pairs found in the bacterial genome. And its function is to allow bacteria to break down and use lactose when lactose levels are high and glucose levels are low. So that's the function of it, and there are quite a few steps involved in terms of what we need to know. So this diagram shows us the section of DNA in the bacterial genome, and the respective genes are in boxes. So we have the regulator, I, which produces a protein, which acts as a regulator. We have the promoter, P, which is like a binding site for a polymerase enzyme to synthesize mRNA. We have the lac operator, which acts as a binding site for the regulator. This will become clear shortly. And then we have the structural genes, which are lac-Z and lac-Y. So what they code for are two enzymes which allow the bacteria to break down lactose. Lac-Z codes for an enzyme called beta-galactosidase, and lac-Y codes for an enzyme called lactose permease. We'll look at a bit more about what they do later on. So, firstly we need to know what kind of conditions affect the lac operon. So let's just say we've got normal conditions with plentiful glucose and no lactose in the bacteria's surroundings. So what happens is the regulator is going to be converted and read and converted into mRNA. So converted into an mRNA strand, and then that will get converted into a protein. So that's our regulator protein, which we'll call I, as that's the letter that you need to know for this. So that is the regulator protein. So what happens is, in normal conditions, the regulator is able to move and it binds to the operator region, like so. So we can see there that that regulator protein is bound to the operator. Now this is important, and I'll explain why. Because when we look at this diagram, we've got two terms we need to know here, and one is upstream and downstream. So what that means is, when this section of DNA is being read, it's read in this direction. Okay, so this end is upstream, and this end is downstream. So what that means is, if there's an RNA polymerase enzyme moving along this section of DNA, it moves this way. Okay? So the problem here is that because the regulator is bound to the operator, RNA polymerase cannot bind, move along this section, and it can't synthesize mRNA from lac-Z or lac-Y. What that means is that the enzymes beta-galactosidase and lactose permease won't be synthesized. Okay? So under normal conditions, when I say normal I mean low lactose conditions, this is what happens. So these enzymes can't be converted to mRNA and then into proteins. But what happens in high lactose conditions is slightly different. So what happens is, if there are high levels of lactose, lactose will come in and it will bind to the regulator protein. So lactose binds to the regulator protein and that changes its shape. So I'll draw that on here. So lactose is bound to the regulator, meaning its shape has changed. So what that means, is very important, is that this can no longer bind to the operator. So that because of its shape change, it can't bind to the operator. We call this the inducer regulator complex. So what happens is, because there is no regulator protein bound to the lac operator, RNA polymerase, so RNA polymerase is able to bind here, move along this section, And what that means is that these genes for LAC-Z and LAC-Y, which code for the enzymes, these can now be converted into mRNA. So converted into mRNA and then can be converted into protein. And then they will act as enzymes to help break down lactose, and then it can be used by the cell for its functions. So in high lactose conditions, that is what happens. Just a point to mention is that lactose, in this case, is known as the inducer. It brings about the transcription and translation of these enzymes by its presence. Okay, so that is the LAC operon. So you need to be familiar with that process and what happens in the LAC operon. So thank you very much for watching. Please like, share and subscribe. If you have any questions, please leave them in the comments, or you can follow me on Twitter at Rushcliffe Bio and tweet me as well. Thank you very much.